So we are very excited for the day. I'll just introduce myself briefly for those of you who may not know who I am. My name is Allyson Berent. As Dr. Yu just mentioned, I am first and foremost the mother of Quincy, a now 4-year-old little girl with Angelman syndrome. Thank you. I am also proudly the Chief Science Officer of FAST. I think I'm going to just use the big mic because it's louder. And I am the Chief Operating Officer of Genetics Biotherapeutics. So I am a veterinarian, but that went from one of the biggest parts of my life as a workaholic to one of the smallest parts of my life the day that Quincy was diagnosed. So that has been a whirlwind and has been a blessing in disguise for me. So the presentation mode is not on. You're going to do that. Okay, great. So for me today, really what I want to talk about, and I've given some parts of this talk years past, but I think it's really important that we all remember what Angelman syndrome is. And you're going to hear some amazing talks today, as you just did, about what we can do to treat it, which is one of the best parts of today, and always is that we're all here to know how are we going to potentially improve the lives of our children and our loved ones. But it's really important that we understand what it is and that we understand the genetics behind Angelman syndrome. And I think if you understand it, you have power. And knowledge is power. And as parents, we know that we need to understand things when we're making educated decisions about what we want to do with and for our children, if we want to do something therapeutically for our children. And so it is my job to make sure that every one of you feel very comfortable understanding the genetics, because it's complicated. But if you really bring it down to schematics, I think it's very digestible. And so I'm here to answer those questions. I'm here to try to explain that, and hopefully my PowerPoint will work shortly. And hopefully you feel comfortable just asking me questions if you don't understand it and it's not digestible. So once this is done, then we will be good for the rest of the day, I promise. So the first thing I want us all to remember is that Angelman syndrome is across the board, it's a disorder of a single gene called UBE3A. Some of our kids have multiple genes affected if they have a deletion, but all of our children have the UBE3A gene affected. So when we think about upstream and downstream therapeutics, which I'm going to talk about, we're going to talk about upstream in the way of thinking how can we replace or how can we activate that gene so that the gene that they're missing is now working. And then when we think about downstream therapeutics, we're going to think how can we address the symptoms of Angelman syndrome? How can we think about not necessarily the genetics, but how can we think about if my child is having a seizure, what is going to be the best medication for that form of seizure? Or if there's an abnormality in the neuron that is because of the missing gene, can we fix that abnormality in the neuron or the brain cell? And so I just want to go through with you all those different options because we are incredibly lucky, not for PowerPoint, but we're incredibly lucky that we have a lot of options on the table that are really available or hopefully will become shortly available for our children. And so I want everyone to keep in mind that we're going to talk a lot about the different therapeutics today. And the most important thing for everyone to know is that when every researcher is talking, when any pharmaceutical or biotech is talking to you, or us as a foundation is talking to you, the number one priority is safety. And so we will never take time over safety. We will always think about safety first. And we hope that's very clear. We all feel a race against time, and we're very lucky that our children don't have a degenerative disorder like Mila. We're very lucky. But we also need to realize, and there'll be a discussion later on today in a roundtable, which I know we had a lot of questions about that therapeutic window. What does that mean for my child who's 25? What does that mean for my child who's 10? What does that mean for my child who's 3? And those are questions that we want to be able to answer, but unfortunately we can't. So we can't answer that because our children are not mice. Our children are humans. And until we put some of these therapeutics into humans, we're just not going to know the answers to those questions. So we need to keep that in mind as we can do best guess based on rodents, but our children learn. They have a much more complex brain than a rodent, and we cannot judge the learning of a rodent, particularly a mouse. A rat may be a little closer, as we can in a human. So keep that in mind and take everything there with a grain of salt. This is still not working, so what am I supposed to do? Yeah, what should I do here? Just end show? Yeah, that's okay. I'm going to keep talking because I think there are a lot of questions from the audience. Yeah? You're looking at me like you have something to say. Yeah, are there more questions for Tim? That actually is fine if I didn't see any more hands, but if there, you probably prefer to hear from Tim more than me, so that's also totally acceptable. He's much more handsome than I am. Anybody have more questions for Tim? Okay, then I'm going to just keep going with your questions that we had on Facebook that I think we're going to try to answer all of them this afternoon, but I thought I can address a lot of them myself because I think some of them are unfair questions to ask our scientists and our pharmaceutical companies, and I don't think they're going to be able to answer them for you. And one of those is about therapeutic windows, and they'll do their best to answer that. But my quick answer with therapeutic windows is we don't know. And so we all want to know that answer, and what we do know is that if we can rescue things in an adult with one therapeutic, we might not rescue it in an adult with another therapeutic. And that's why everybody talking today has such incredible value, because downstream therapeutics may help seizures where upstream therapeutics may help. I saw a thumbs up, but I don't feel that there's a thumbs up. And upstream therapeutics may help motor function. And so there may be a cocktail and a combination that we need to think about for our kids. So every approach matters. Every approach is important. And it's an unfair question to say, will my child benefit from this because they're an adult? Because as far as I'm concerned, every child will benefit from hopefully everything in some way. And whether younger kids benefit more than older kids, that's possible. But it doesn't mean that all children won't benefit, and they all may benefit miraculously. And so all we can do until we know the answer when it's in humans is hope that that's the truth. And I think that's going to be the best answer to that question. I'm dying over here, a very slow death. Okay, so that was my one thing. The other questions that we had were that people asked was which therapeutic is the most promising? And again, I also think that's an unfair question because we cannot answer that at all. They all look incredibly promising, and some look, dramatic and some look less dramatic, but the less dramatic ones may be more dramatic in humans than they are in rodents. And again, we only have rodent data, and a rodent is not a human. And I suspect our human brains and the brain's ability to learn with time is going to show much more improvement than a rodent, because we're testing very, very subtle changes in rodents that really are often not translatable to humans. So that's my opinion on that. Okay, we're still not there yet. So, that's not the doc that we want. Number 2. Oh, okay. Can you just copy the second one because we have a break after this, and then we can do the rest of them later? Would that work? Okay. Yeah, we could do the break now. Wouldn't that be good? Okay. I think there it goes. It was working before. Why isn't it working now? That's so weird. Okay, well, I'm just going to start the presentation, and I think I have it memorized on my own computer, and then we'll go from there when they change it. So when we think about the genetics of Angelman syndrome, it's important for all of us to realize that when we talk about the disorder and we're talking about the genetics of that, we're talking about a single gene. But that single gene is, again, in deletion children, may have more genes around that. The one gene that we know has the most impact for Angelman syndrome is called UBE3A. That UBE3A gene and Angelman syndrome in general, and having a problem with that gene, has been estimated to affect somewhere between 1 in 12,000 to 1 in 25,000, even up to reported 1 in 62,000 children in the world. We don't really know. What would that translate to? That would translate to about 350,000 people in the world. If we look at populations, that would translate to, somewhere around 25,000 in the United States. But oh, wait, I'm ready to go. Oh, amazing. Okay, thank you. Clicker's not working. Just saying. Now it's working. Okay. Okay, so like I just said, nope, sorry guys. Clicker's not working. Okay, I think clicker's working. All right, thank you. So like I said, it affects the 15th chromosome. This is a karyotype, and this karyotype shows you that there are 46 chromosomes. There's 46 pairs, 46 chromosomes there, and of those, it affects the 15th. So there's one for each number. So we have 23, and then we have one from mother and one from father. So there are 46. It is on the 15th chromosome that the UB3A is sitting. And like I said, it can affect anywhere from 1 in 12,000 to 1 in 62,000 births. Each chromosome is made up of various genes, and those genes are coded to produce different proteins, and those result in the proteins that we make in our body result in different bodily functions. So when we think about the schematic of Angelman syndrome, hello people, you're killing me softly. Not working. Okay, we have pairs of chromosomes. Okay, so this is the 15th chromosome, and we have one copy from mom, called the maternal, and one copy from dad, which is called the paternal. And of those copies, okay, this isn't working. So should I just say advance, or what are we going to do here? Yeah, I have lots of animations, so that's going to be really distracting. Advance, okay, I'll just say advance, okay. So this is a schematic of those chromosomes, so you can see maternal and paternal. And we have the UB3 on the maternal, and advance. And we have a stop on the paternal, advance. And we have something called the antisense that is sitting over that stop. And when it is sitting over that stop, what that means is that, advance, that we have an imprinting gene. Can someone fix this, because this isn't going to work? Thanks. That we have an imprinting gene, and that means that that paternal gene is silenced. And if that's silenced, then we don't have a functional working copy of the UB3 gene that's sitting on the paternal allele. Unfortunately, in Angelman syndrome, the UB3 gene on the maternal copy is also silenced. So if it's silenced, we now have nothing working on the mom side, and we have nothing working on the dad side. But the good news is that there is a copy on the dad side, it's just silent. And so if there was ever good news about Angelman syndrome, the good news is that somebody thought if there's a problem on one, maybe we should create one on the other side that might be functional. So that was my saving grace the day that I learned Quincy's diagnosis, is that we can activate a silent copy of a gene. Mila doesn't have a functional copy of the gene on either side, but our kids have a functional copy of the gene on the father's side. How can we exploit that mechanism and make that work for our kids? And that was the day that I met Art Baudet. Advance. So what happens if we disrupt the UB3A gene? If we disrupt that gene that's on the mother's side, when we don't have a functional copy on the father's side, we're going to have Angelman syndrome. And Angelman syndrome can present in a demographic in different ways. So we can have deletions, which is about 70 to 80% of our kids have a deletion. So if you look at that first column, basically you can see the first is normal, the second to the right of that schematic is shorter. That means that they're missing a chunk of DNA. And if they're missing a chunk of DNA, which is made, genes are made up of DNA, then they're missing that gene completely. They don't have a chance of that gene working. So they only have the option of replacing that or activating the father's copy. The next is a mutation. A mutation is where they have the gene, it's present, but something on that DNA transcript, all of the letters of DNA that make it work, is abnormal. So they had an exchange where one letter got replaced for another letter. And it can be that simple that that exchange makes the whole gene not make a proper protein. And if we can't make that proper protein, we have Angelman syndrome. The next is UPD. That's where the child has actually 2 copies from dad and never got mom's copy. So during development, mom's copy was kind of replaced by a second dad's copy, and they're both silent. So we have no functional UBE3A gene on the mother's side, and we have 2 silent copies from the father's side. And then finally, something called an imprinting center defect, or ICD. And that's basically where everything looks gorgeous. So then you're like, what the heck? Why does it look so good but act so bad? And that's where the imprinting mechanism, in order for that gene to be read, is abnormal. And so the gene itself is perfectly healthy, but in order for that gene to be properly read, that is abnormal. And so the ultimate similarity between all of these is what we deal with every day, is Angelman syndrome. They don't have a functional copy of the UBE3A gene. No matter how it panned out on how they got there, they don't have a functional copy. Advance. So what we think about the UBE3A function, how does that gene function? Advance. We know that the protein is vital to life functions, and we know that it controls speech. We know that it helps to control movement. We know that it helps to control this. Might work. Awesome. No, I hate to tell you, not working. Nope. Not working. I'm going to give them all to you because none of them are working. Okay. Advance. So provides instructions for making this protein, and how does that protein affect our body? Advance. So the protein, back up. No, back up. Oh. Back up. Thank you. So the protein is an enzyme, and that enzyme is incredibly important in the brain. And what that enzyme does is it's kind of like the vacuum cleaner. So it tags other proteins in the brain, and it tells those proteins whether they should basically stick around for a little longer or they should be broken down. And so we don't want garbage accumulating in the brain, and we don't want all of those proteins in there, because we make proteins and then we degrade those proteins so that we make new proteins, and our brain can function in a way that's very organized and very clean, and we only make what we need when we need it. Well, if we have a protein that does not actually is not broken down and is not discarded, then we have an accumulation of proteins. The good news is that accumulation of proteins is not degenerative. So there are many accumulative protein disorders that are degenerative, and they actually, cause neurologic loss and, holes to be developed in the brain, and really the loss of neurons. We don't see that with Angelman syndrome. That's the good news. This is a biochemical issue where we have too much protein building up in the brain, and there is a lot of noise that happens. And I always kind of use this example, and I feel like I've heard other people now use this example, so I think that means that it makes sense to us as parents. But when you have an accumulation of proteins and all these signals happening in the brain at the same time, I kind of always think about Angelman syndrome as like we're driving in a car and it's pouring rain, and we have to turn the windshield wipers on so we can see, but we kind of see between each wipe because it's going so fast because it's pouring. And so you kind of want to pull over because you can't see that well, but you know that you can kind of see, and you should probably just keep going because everyone else is going. And so you're following the people in front of you, and you're doing your best, but you're staying far behind because you don't want to hit them. But it's hard to see. Well, that's what our kids live with every day. They have their windshield wipers going super fast, and there's a lot of noise. And so what happens when there's a lot of noise is they have to weave in and out of that noise in order to accomplish the simplest task, even just walking. That there's a lot of noise going on for their brain to say, I need to take a step, and the rest of you need to be quiet for a minute while I think about taking that step. We, neurotypical humans, don't have that. We can control our movements, and we can control what we need to because we can inhibit things at one time and we can activate them at another. Advance. So when we don't have this removal, we don't have normal cells. If we can get that removal to occur, we would have more normal functioning cells. Advance. So without the ability to remove that, we ultimately have this buildup. Advance. And that is localized in this schematic, which I give credit to Ovid because I love this picture and I always use it, but I always give you credit. I think this is a really great depiction in my head of what makes it easy to understand Angelman syndrome. So for me, if I look at this picture shows that we have a neuron from the top talking to a neuron from the bottom. So that's called a presynaptic and a postsynaptic neuron, and they talk to each other. And they talk to each other through those proteins, and you can see all those little dots. They look like, little ants. And those are the proteins that are talking to the nerves that make things coordinate, and they make functions happen. So I need this nerve to tell this nerve what to do, which will talk to my muscle and tell my muscle what to do. And so if we have too much accumulation of proteins, that noise is that creates an abnormality in that pathway, that it's not quite as fast as we want it to be. And so what happens is that we can have some of the proteins that we want there to be accumulated back into the neuron, and we don't have enough of them where we want them. And then we have some that we don't want there to sit there for too long and create, symptoms that we don't want to have there either. Advance? So the lack of UBE3A and the lack of the UBE3A protein ultimately results in the loss of something called tonic inhibition. And we all talk about tonic inhibition. We're going to hear a lot more about that when Ovid gives their presentation later with their STARS trial with carboxydol. But what tonic inhibition is exactly that. It's creating an environment in that synapse or that junction of those two nerves where we can tell the second nerve, I want you to not do something, and the next nerve next to it, I want you to do something. So it creates this coordination, right? So we want to say, okay, I'm going to take a step, and my forward step is going to lead, but my back leg is going to relax. But now I need to go forward. So now my left leg is going to lead, but my right leg is going to relax. And that's a activate, inhibit, activate, inhibit. And that's what we do when we're talking, when we're walking, when we're thinking, when you're not allowing me to advance my slides, and I'm frustrated, but I'm doing it anyway without, really complaining about it because I'm talking to an audience. I'm getting that out of my head because I'm focusing on what I need to do up here. So that is tonic inhibition. I'm allowing myself to say, okay, I have to go to the bathroom, but I'm not going to go right now because that would be wrong, but I'm going to finish this talk and then I'm going to go to the bathroom. That is what I can do to control myself because I can have tonic inhibition. So if we don't have tonic inhibition, all of those things just keep happening, and it's really hard to tell ourselves what to listen to, what not to listen to, what to focus on, what not to focus on. Advanced slide. And it results in the symptoms of Angelman syndrome. What are those symptoms? The universal lack of speech. Well, we can't coordinate the hundreds of muscles in our tongue and our face in order to make a word. And if we can't inhibit and activate when we need to at rapid speed, then how are we going to speak? So one part of the universal lack of speech may just be that motor coordination. And that may be misinterpreted as cognitive deficit. And that's unfair, because if you don't give them the ability motorically to do something, doesn't mean they don't have the cognition to do something, right? And so we misinterpret cognitive impairment maybe with motor function. And maybe it's a combination of things. We don't really know. And so they have a universal lack of speech, life-threatening and potentially debilitating seizures, significant developmental delay in all of our kids. And these are universal: ataxia and dyspraxia, unique behaviors and characteristics, that happy demeanor, unable to live an independent life because they can't do everyday functions that we can do with having this functional gene. And so what we do know is that there's a very significant clinical unmet need in the Angelman community. And everyone sitting in this room realizes that. And the good news is everyone sitting up front, our pharmaceutical partners and our academics also realize that. And that's why they're here. Advance. So what's our roadmap to success? Advance. So we want to investigate new genetic strategies, new therapeutic strategies, and this is FAST mission. FAST mission is to look at all of the different strategies that are out there, figure out what are priorities for funding, and make sure that we bring that to our kids. So whether that's gene replacement or protein replacement, whether that's paternal gene activation, those are both options. Advance. Identify symptomatic treatment, so downstream therapeutics like I just talked about. Advance. And we want to prepare for clinical trials. We want to prepare for all of the work we've been doing in labs and academic institutions. We want to then be able to translate that. And how can we translate that from, as Tim said, from the bench to the bed? And there's a bridge that needs to get there, and that's a huge bridge. And 4 years ago, when my daughter was diagnosed, we weren't on the bridge. We were on the bench. And today we stand on a bridge, and that's a huge difference. Huge difference. And what does that mean? We need to fund a lot of stuff to cross the bridge and get to the bed. And that is really, really critical right now. Advance. So when we think about gene modification therapies, advance, we think about gene replacement therapy or protein replacement therapy. So we take maybe a viral vector, as you can see there, and we fill it with UBE3A gene. And we deliver that into the neuron of the brain so that now the protein is made. It's the trickle-down effect, and all of those issues at the synapse and how it distributes like an umbrella may be benefited. And those symptoms may be ameliorated if we can just replace that gene. Advance. What if we could activate the paternal allele? What if we can just turn it on and say, okay, we have one sitting there, it's silent, it's waiting for someone to do something about it. Can we do something about it? And that's where we might be able to activate the paternal allele. And so we have a lot of partners working in this space. For gene therapy, we have the University of Pennsylvania program. PTC Therapeutics has a program. The University of South Florida is working with PTC, so Dr. Ed Weber and Kevin Nash. UC Davis is working on a gene therapy program that you're going to hear about later today. So all super exciting. They're all here. What if we want to turn on that paternal? I always call it, we're going to stop the stop, right? Because we're stopped on the father's side, but we want to stop that stop. I'm all about double negatives. Let's turn it on. And so how do we do that? Well, as Dr. Yu just mentioned, we can use antisense oligonucleotides, and we can stop that antisense so the gene can be read. And we have a talk later this afternoon that Dr. Dindo will give about antisense oligonucleotides. We have genetics, we have Roche, Biogen, Ionis. They're all in the room. They're here because they're interested in developing antisense oligonucleotides. So we have a lot of people working at this, and if not all, are going to figure it out, and that's super exciting. And artificial transcription factors. Dave Siegel's in the room. He's going to talk about a lot of his work, and I'll mention it shortly. From UC Davis, he can activate the paternal as well, and he has showed that now for a couple of years. And then there's some other newer projects that we're funding that are much more in the proof of concept phase, but we're not stopping until we figure out the right approach. Advance. What about downstream therapeutics? Like I said, well, those are still super important, right? Because what if we have a genetic rescue, but it doesn't rescue seizures, or it doesn't rescue motor, or it doesn't rescue, some constipation? We still need to think about how we're going to address all of those issues. So it's holistic on our approach. And so downstream therapeutics, we have Ovid here looking at GABA replacement therapy, disruptive nutrition looking at ketone supplements. NYU, Christine Alberini just spoke, and she has an amazing talk she's going to give later this afternoon, looking at other novel compounds like IGF-2 to help that synapse, and some anti-inflammatory regenerative type of stem cell therapy by Neuralstem that's looking at downstream therapeutics as well. Advance. So how can we help prepare for clinical trials? That is our number 3, and that's where we are right now. Well, we had papers that were published looking at the disease burden and the clinical unmet need in Angelman syndrome, and we know it's there, and the FDA knows about it. We have a disease concept model that was just presented yesterday by Roche that they're going to talk a little bit more about later this afternoon. Super exciting. Really, they're going to share with you a lot about what is important to families, because all of you participated, or many of you participated in that study, telling them what's important to you and your child. We have NIH funding of 8 years of data, of natural history data. Thank you to all of you in the room who participated in that back in 2006 to 2014. All of that data is super important to look at the natural history of the disorder so we can look at changes. We have an alliance, a consortium of outcome measure and biomarkers that we're looking at in order to make sure that we look at the right tests in order to know when therapeutics come to our kids that we can show change, because we can't approve a drug if we can't show change. And so we need to know, what are our kids starting with and how can we show change? And then finally, the Global Angelman Registry, which Megan Cross and her team will talk about this afternoon, which is super important that we all register, because that's where we're going to have a lot of that natural history data put in. And that is very, very important. Advance. So gene replacement therapy, advance. Clinical trials are underway in humans and for other neurologic disorders, not for Angelman syndrome, but it doesn't mean that we're that far behind. Here are just some examples that I've shared with you in years prior, but it really is a new era of gene therapy. And gene therapies are in numerous clinical trials, and PTC Therapeutics is going to be talking about that today, as well as Jim Wilson, about the Angelman gene therapy programs. So I'm not going to talk too much about that, other than to let you know that there have been many that have preceded us with gene therapy, and there are many clinical trials that are happening. So we are really just leaning off of a lot of their experiences in terms of safety and hopefully distribution and efficacy. Advance. What we do know is the first gene therapy was approved in 2017 in August, so that was amazing because that really led the way for many more companies coming into the space, and that's incredibly important. And that was for inherited blindness. Inherited blindness, some of these kids never saw anything, saw little to nothing, had gene therapy, and 4 days later could see a hot air balloon in the sky. Never saw anything before. So I have a really hard time believing that it's ever too late for anything for any child, because that's an 8-year-old child that had no vision that now has excellent vision. And if any of you saw America's Got Talent, you saw the child that got treated with gene therapy on there who now has vision, and he did not have vision before. So you can't tell me that there's ages that we need to put a line in the sand of when things are possible, because until you put it into a human, anything is possible. Advanced? What's effective in Angelman syndrome? Well, Dr. Weber really laid the groundwork for gene therapy in Angelman syndrome in 2011 with his publication looking at the fact that you can activate the you can replace the UB3 gene in the brain and actually recuperate some of the symptoms of Angelman syndrome in an adult mouse. And that was the first time anyone was like, what? You could do that? People used to think you needed a brain transplant for gene therapy. Like, that was actually stated, apparently, at some scientific conferences. No, you don't. You just need a really good virus. And we're really lucky that we have people involved in our programs that invent these really amazing viruses that can deliver this gene. Next. So we need to know about those vectors. We need to know where they're delivered. We need to know how they're delivered. And we need to be able to think about what's the safest and the best approach. Next. We have 3 programs that are actively we're actively involved in right now, and obviously we'll talk about all 3 of those throughout the course of the day. Next. And this is just some data that I'm going to skip, but just to show you that brown, all you need to know, because I know data is overwhelming, all you need to know that brown is really good. Okay, that's all you need to know. That brown is really good. Next. Okay, this is something, I'm not going to steal any thunder here. This is Dr. Joe Anderson's work, and he's going to talk a lot about this. This is another disorder called metachromic leukodystrophy. He's going to talk about his hematopoietic stem cell gene therapy work, which I am so psyched about, and I cannot wait for him to present to you guys about this, because you're going to be blown away. So I won't share any data here. Let him do all of that. Next. Now, let's talk about paternal knockdown. Next. Let's stop that stop. So in 2011, it was published by Ben Philpott's lab that you can actually activate the paternal allele with a medication called topotecan. Next. Antisense oligonucleotides were first published by Arpo Det's group in 2015, also published activating that paternal in 2013. And that was the proof of concept that really was the launching pad for many people in this room to think about how they can use that in order to bring forward therapeutics for Angelman syndrome in a way that's meaningful, effective, and quickly. And that really was the groundbreaking work that all of us look to and bow to, because we're so grateful that that really opened up the world for activation of the paternal in a way that's therapeutically translatable. Next. Artificial transcription factors, we saw that published a couple years ago by Dr. Dave Siegel's lab. Next. And basically he was able to show that you can really get distribution throughout the brain of paternal activation. Next. So this is just some images. Again, that red, really good news. Okay, paternal activation, the gene is on. Next. Red, really good news. Paternal activation, the gene is on. That's really all you need to know. Next. Okay, so downstream therapeutics. So we are looking at downstream therapeutics. Ovid is going to talk all about their STARS trial with gaboxidol. Super exciting. This was published in 2012 in animal models, and they rescued a ton of symptoms in adult animals. And so what that showed us was that age may matter for one therapeutic, but not for a different one. They rescued different symptoms in adults than gene therapy rescued in adults. They were different. So that doesn't mean that one is better than the other. It means we might need a cocktail. And so we need to look at all of this. It's all very important. Next. Ketone supplements, Disruptive Nutrition is going to talk to you today about how ketone supplements can affect so many parts of the brain. Again, it's downstream therapeutics, neuroinflammation, ketones improve cognition, anxiety, seizures. There's so many things that are downstream that ketones can have a major impact on. So they're super excited. Next. IGF-2 ligands. Dr. Alberini is going to talk to you about that. How does that drug affect the synapse? Hi, Marina. She just wanted to say hi. Hi, Marina. Hi. Next. So what's our primary goal? Each therapeutic target can benefit kids in different ways. Different individuals may benefit, and we need to think about the combination of how they're going to benefit with each one. Investigating each one thoroughly is what is our obligation to all of our children, and that will never change. We want an ultimate symptomatic cure as close as we can to treating all of the symptoms. Next. So bringing therapeutics to humans, and we're going to be done very quickly, but if anyone remembers that was here 2 years ago, I kind of ended my talk here. So next. This is going to be a lot of nexts just for you guys in the back. Okay, so this is just a roadmap that you need for drug approval at the FDA. Next. This is AAV therapy. 2 years ago, this black line is where we were in drug discovery. Next. 2 years ago, this is where we were in ASOs. So it was AAV is 1, ASOs is 2, ATFs, artificial transcription factors, is 3. Next. Downstream therapeutics, gaboxidol, is 4. They were pretty close to announcing a clinical trial starting soon. And 5 was our stem cell program for gene therapy. So we were really in a basic research proof of concept. Next. And then we needed to think about, we're entering that preclinical phase of IND. What do we need to look at in terms of outcome measure and biomarker testing? Next. Next. So where are we today? Okay, so pay attention. The colors are going to change and a lot of nexts are going to happen back there. Next. Okay, this is where we were 2 years ago, just to remind you. Next. This is where we were 1 year ago. Next. This is where we are today. Next. For ASOs, this is where we were 2 years ago. Next. This is where we were a year ago. Next. This is where we are today. Next. ATFs, this is where we were 2 years ago. This is where we are today. Next. Downstream therapeutics, this is where we were 2 years ago. Next. This is where we are today. Thank you to Ovid. And we have other downstream therapeutics like ketone esters now who have started their clinical trial as well. So we have 2 things in clinical trials right now. Next. And we have others that are approaching. Stem cells, this is where we were 2 years ago, and this is where we are today. Next. So that box, that red box, is filling up really quickly. And I expect next year we're going to cross that line in many more areas. Next. So we need to help prepare for clinical trials. Patient-centered outcome measures matter, and your participation is absolutely key. We need to think about what improves the quality of life of our kids, and this is where your input is absolutely necessary. So we know speech matters. It matters to most of our kids. Improved motor function, improved sleep, improved seizures. That matters to our kids. And we're here to make sure that any therapeutic, the only thing that matters is if it improves the life of our kids. This is not about anything other than that. Next. Changing the biology is one thing, but we have to be able to measure change. So your participation in allowing us to help measure change is really important, and we're going to talk more about that later this afternoon. So biomarkers matter. What's happening in their brain, and how do we translate that into what's happening on their feet? How does the little girl with an EEG on her head translate to the little girl who's walking? How does that translate? And that's what the FDA cares about. Next. What matters to patient and caregivers? Next. We can just skip this slide. Next, for the sake of time. Global Angelman Registry. Megan's going to talk about this. Pay attention, you need to register. You'll get a sticker, and there'll be a donation made in your honor. Next. The Outcome Measure and Biomarker Consortium. We're working really hard to figure this all out for you guys, and we love how much you have participated. So please keep it up. We are so grateful to every family that has participated. Next. Epilepsy matters, communication matters, motor skills, gross motor matter. Next. Those are what's important. So bringing therapeutics to clinical trials, gene therapy, next. Turning on the paternal allele, next. And downstream targets. We have a long way to go for each of them, but it's a short area in that red box. But it's just a lot of work in a short period of time that we need to do, and we need your support. Next. So our roadmap to success, next. Investigate new therapeutics, identify treatments, prepare. We're doing all of those things thanks to everybody in this room, and we cannot be more grateful to every dollar that has been raised by all of you in this room. Next. In summary, therapeutics are surrounding our community. AS is clearly a promising disorder to treat, or we wouldn't have so much representation here. Knowledge is power, and you need to understand what you're getting yourself into. You need to understand Angelman syndrome. That will help you make the best decision for your child. No one's here to tell you what is right for your family. All we're here to do is give you options and opportunities. Next. I want to thank everybody here for, particularly the families and our friends, for all of the support for our community, the Fast Fire team who believes in all of us, and we would not be anywhere near where we are today if it wasn't for the work of our amazing fire team who is all sitting here and is going to be speaking today. But we launched 2 years of incredible research. Thank you to the Manier-Lapostal Foundation and the $5.8 million grant that we got 2 years ago. And we have come to this point to stand on a bridge because of the unbelievable work that has been done by our Fast Fire team and our partners. And we are so grateful to them. Our pharma and biotech partners, every one of them that is sitting here, we're so grateful for believing in our kids and believing that treatments are possible, because if you didn't think you could see therapeutic benefit, you wouldn't spend the money. We know that. And so we know that you believe in our kids or you wouldn't be here, and that makes us so ultimately excited. Next. Next. Oh, okay, that's it. Thank you very much, and I'm happy to take a couple questions. Any questions? We can take a couple quick questions, and then we'll run to the break. Any questions? Raise your hand high. No. Okay, come find me if you do. Oh, there's one. Hi.
Allyson, thank you so much for talking to us today. And I see a strong analogy in the issues with the AV in that you saw a challenge and you blew right through it and you got through everything. So and it's just an analogy with everything that you and Paula have been doing and the board the last few years. So you took that challenge, blew right through it, and you accomplished what you needed to do. So thank you.
Oh, thank you very much. That's very kind.
Technical question. In neurotypicals, in neurotypical humans, is the paternal gene always a stop? Do you gather my question? So is it just in AS patients that it is a stop at the paternal level?
That's a great question, and I apologize if I didn't make that clear. So the question, for those of you that couldn't hear it because it's not that loud coming from over there, is do all humans, and really mammals, have a paternal stop on their paternal allele of chromosome 15? And the answer is yes. I have one, you have one, all of us have one, but we only need one functional copy. So I am working, I'm working and tonic inhibiting over my mother's copy of my gene. And so our kids don't have a functional mother's copy, but just like us, they have a silent father's copy.
Thank you.
Any other questions? Okay, everyone go have a break. Back in 10 minutes.